Okay. Hello everybody. We're gonna back with another lecture review. And this time we're going over lecture 23, if I'm not mistaken. Which is gonna be covering electron transport. Woo. As I said before, my voice is finished, so cut me some slack, please. But let's go ahead and get into this. So we have already talked about the Krebs cycle, citric acid cycle, right? And it produces 3 NADHs and one FADH2, right? So that's very important, right? These electron carriers end up going through the the electron transport chain, which ultimately reduces oxygen into water, right? So O2 in this state has a oxidation state of 0, and then over here it's -2. We're dropping off 2 electrons on the oxygen, right? And the main thing here to note is that NADH goes into complex 1, and FADH2 goes into complex 2. And what you notice is something cool is that succinate dehydrogenase is the enzyme here. So this enzyme is actually embedded in the membrane, right, of the mitochondria inner membrane specifically, and takes part in citric acid cycle and in electron transport chain. So I already talked about before, right? How, we basically have NADH going in here, right? And then that NADH pumps protons from complex 1, then goes to complex 3. The electrons go to complex 3, pumps proton, complex 4 pumps proton, right? So complex 2 is different because FADH2 drops its electrons at complex 2. Complex 2 is not a pump. We're skipping complex 1. Pump from complex 3, pump from complex 4, and then we've made our gradient. So there's actually less pumping going on with complex 2 than complex 1. And then when you take those H pluses and then go down the gradient, right, you produce ATP. Right. So this idea that, electrons pass through a series of membrane-bound carriers. Membrane-bound just means that they're bound to the membrane, right? And they're basically attached to these complexes, right? So like complex 3 and complex 1. So there's a couple types of transfers that occur in oxidative phosphorylation. There's direct movement of electrons, like the transfer of electrons from one place to another, right? There's the transfer of a hydrogen atom, right? And this is what you usually get with like NADH, generally speaking. And then there's transfer of a hydride, right? And then when we talk about reducing equivalents, we're talking about a single electron, right, equivalent transferred in an oxidation redox reaction, right? So like a single equivalent of electrons, similar to like an equivalent of like acid base, right? The equivalent of electrons, right? So how much is required to reduce a whole species, basically, right? So electron carrying molecules, there's a lot of them. So there's NAD, so vitamin B3, right? So B3. There's flavoproteins. This is like a riboflavin-based stuff, right? So like B2. Ubiquinone is actually Q, so this guy actually carries stuff from complex 1 to complex 3, and also complex 2 to complex 3. So coenzyme Q otherwise. And then cytochromes, so cytochrome C specifically carries from 3 to 4. And there's iron sulfur proteins that also like are within the complexes, basically. Those are the main electron carrying molecules that you need to know. And here you just have a massive table of the redox potentials for a lot of these things. So what you should notice is that starting from like ubiquinone, basically, right? Actually, NADH dehydrogenase is the first complex. Basically, what you should notice though is that like as we go down, the reduction potential actually gets more and more. So this is actually how we're able to drive the reaction, right? To actually cause a movement of electrons until we get to oxygen. Okay? We're basically just slowly but surely causing spontaneous reactions until we get to 0.817. We can't just go straight from like this to like 0.817, because what happens is you just lose a lot of the energy as heat, and you just don't harness any of it, which is a problem, right? And here's a nice little image of what this actually looks like. So you can see up here, we're starting at like negative 0.35, right? And then we end up at around around positive 0.8 for the reduction potential. You can imagine that if we go from 0.8, so if we do 0.8 minus negative 0.35, it's like a positive 1.15, right? So that's a pretty big drop in reduction potential, but we can't do it all at once because we're just going to lose all of our energy as heat, and we need to harness the energy. Iron sulfur proteins are proteins that contain iron that are associated with sulfur atoms. And I would say this is not the most important thing for this exam, or just for this class in general. Know that they exist, and they exist in many different forms. So there's also like this Rieske iron sulfur protein. I don't know if I said that right, but that's also a different version that's coordinated with two histidines. But I wouldn't be too worried about this slide, to be honest with you. I think there's better things to memorize. So this is NADH dehydrogenase for bacteria, so prokaryotes, not mitochondria. Just to want to make that clear. But I think the mechanism is like it's very somewhat similar. So it's generally the same idea. But what happens is the NADH gets oxidized. Those electrons go through, and it basically goes through like a steam engine type thing where like as it moves through, protons are pumped. And this is the inner membrane space over here, and this is the outer membrane, which is actually this is the matrix, and this is the intermembrane space. So we're pumping against the gradient that way. So the electron flow, right, basically causes a conformational change, and it causes basically the way that all of these proton transfers work is we have a carboxyl group that gets protonated and then deprotonated depending on the side chain pKa's. So you can imagine that as electrons flow, there's a conformational change that causes the carboxyl group pKa to raise, which makes it more basic, right? And as a result of that, a proton is more readily slapped onto that carboxyl group. And then when the conformational change happens and the pKa gets lowered for that carboxyl group, and it ends up releasing that proton, right? So basically it gets protonated, causes the conformational change, goes to the other side, gets deprotonated because the pKa is much lower on the other side. Here is what ubiquinone looks like, coenzyme Q. It's a nice thing to know. I wouldn't say it's the most important thing. You should note that it's a hydrophobic carrier. It's not like FAD and an NAD, right? NAD plus. This thing is hydrophobic. It stays within the membrane, and then in its reduced form, it looks like this, where it's got a benzene ring, and its oxidized form, it looks like that. Right, so oxidized form, reduced form. And the other thing to note is that it can accept 1 or 2 electrons. So, if you look at coenzyme Q, right, coenzyme Q acts between complex 1 and 3. And all it does is it helps transfer electrons, and actually acts as a bit of a pump itself. So, essentially, what it does is actually it's listed here, right? It accepts a couple electrons from complex 1, so from this iron sulfur complex, this iron sulfur protein, right, in complex 1. And then it diffuses within the membrane between these two complexes, and it dumps off its electrons on the iron sulfur or on the cytochrome Bl, and then it ends up going through the Q cycle, which is a different thing. So the Q cycle, right, is here. So the Q cycle, essentially what's happening is, starting with this quinone, we donate our electrons, and one of the electrons goes up to the cytochrome C, and one of them goes down to this Q and becomes a Q radical. And what happens then is that we go to the second half of the Q cycle, where we take another Q, right? And we now have this, right? And so, like before, one of the electrons goes up to this blue thing, and one of them goes down here, and you end up with this, right? And then what happens is this will then donate its electrons, right? So basically, it's like every 2 Qs that go in ends up making another Q, if that makes sense. So try and rephrase that in a better way. But we have our Q pool with electrons, right? And the QH2, so this has like a couple electrons, right? One of the electrons is going to go up to cytochrome C, and the other one's going to go down to Q, right, to make this Q radical, right? The next round, another Q is going to come in, and then that Q is going to donate one of its electrons to cytochrome C, and the other one's going to go down to the Q radical here. And it's going to make this QH2, right? So the difference between these two is this QH2 versus Q radical. And so now we've sent 2 electrons up here, right, after this second round here. And then we also have another 2 electrons here that can be sent. Right, so that's the main idea I want to get across there. So why is the Q cycle important? It's important because it allows 2 different QH2s to transfer electrons one at a time to the electron carrier, right? So you saw here, this is one QH2 here that's doing the donation, right? And then there's a second QH2 that's coming in. So basically there's 2 QH2s that go in, and they each donate 1 electron, and then they provide their other electron to this radical, right, to make another QH2. So every 2 QH2s makes 1 fully reduced cytochrome C. And here we have another, again, I don't think these are very important. You do not need to know the structures of all of these. Just generally know that they are made of iron in the center, right? That's the main thing you gotta know about these. And these are hemes, right? They're similar to hemoglobin, right? But yeah, I wouldn't worry too much about the underlying specifics of those. So cytochrome C, right, unlike ubiquinone, right, or quinone, which is a, not ubiquinone, the quinone, which is a hydrophobic protein, cytochrome C is water soluble and is an electron carrier. You don't really have to know the exact structure, I would say, but what's helpful to know is that it's a ring of lysines is what helps it dock with cytochrome C1 and with complex 4. This ring of lysines, right, basically allows for some kind of like charge stability interaction with complex 3 is how you can probably think about it. Okay, moving on. Here is the full reaction of cytochrome C, if I'm not mistaken. And you can see that we're basically going from 1/2 of this O2, which is 0 in oxidation state, to -2 over here. So we're reducing. But then we're also going from this reduced, I don't know, let's just say -2 state over here to the oxidized state of 0 over here. So notice the redox reaction always has something reduced and something that's oxidized. Yeah, this is just a nice, it's kind of interesting. How is it determined? The order determined, they determine it using a lot of different things. Like, so the E standard, they reduce the entire chain by emitting O2, and then they try and measure the oxidation rates with each electron carrier. And then maybe they try adding inhibitors. And you should know these inhibitors, by the way. Generally speaking, you should have these inhibitors in mind. Do you have to have them memorized? I would say yes. I would say memorize rotenone, antimycin A, and cyanide. and carbon monoxide. They could be helpful. I have seen them. I think they did test us on that. I can't remember off the top of my head, but that was something that they did talk about in our lecture. If they didn't talk about it in your lecture, don't worry about it, but I'm inclined to believe that they probably will test you on this. And you should also be able to tell like between which complexes they inhibit. That's the main thing. What complexes are being inhibited, right? So like for this one, right, it's going to be complex 1, right? Because going from NADH Q is you have to go to complex 1 for that, right? For this one, NADH to Q to cytochrome B, so cytochrome B to cytochrome C1. So cytochrome B that's going to be complex 2 inhibitor, right? Because we're going from B to C in there. This is going to be complex 2. And then from cytochrome A3 to O2 is just going to be complex 4. Oh, actually, sorry. Did I mess this up? I messed this up. This should be complex 3. Complex 3 and then complex 4. So because cytochrome B and cytochrome C are in complex 3. It's just drawn poorly. And here is the sequence of events. I think the easier way of remembering, I think there's not really an amazingly easy way of remembering it, but you can kind of think about it like the alphabet, right? So we go from NADH to Q, then we go to B to C, and then we're being A is weird because we go from A to A3 and O2. And this order has been confirmed by all 3 approaches. I would try and keep in mind which complexes these things are in. So generally speaking, it's around like 1 or 2 per complex. So like, let's see here. You've got Q, cytochrome B, C, and C1. Or B and C are generally together, and then you've got your other cytochromes A and A3. But these are all generally together. These 3. And then these 3 are generally together. And then O2 is on its own, basically, with complex 4. And here you go. We've got more of this again, like I talked about before. I'm not going to go over this again. But if you just some reduction potential, then you can kind of tell that as we go down, the reduction potential increases, suggesting that it's energetically favorable to do that reduction. I don't really think this is that important, to be honest with you. So it's just about separating different complexes of it and how they do it. They basically just lyse the mitochondria and then take parts of the cell wall. Basically it. Here you've got some different weights. Again, don't think it's that important to know these exactly. You should know these names though. So NADH dehydrogenase, succinate dehydrogenase, cytochrome C autoscleroreductase, cytochrome C, cytochrome oxidase. You should know these names generally. You should also know what groups they have on them, generally speaking. These are just things you got to memorize. So this is actually kind of cool, but it's basically this idea that they can form respirasomes, which are just like these complexes that contain all 3 complexes. So complex 1, complex 2, complex 4, and they tend to provide a capacity for electron transportation, or they end up limiting production of ROSs, because if you're under too much stress, you will end up producing reactive oxygen species, which will actually completely tear up your body. And we've already talked about this before, but cytochrome C and ubiquinone readily diffuse. So cytochrome C from 3 to 4, cytochrome C from 3 to 4, and then ubiquinone goes from 1 to 3 and 2 to 3. Here is just that math I talked about before, right? Where you're taking the final and minusing it by the initial, right? So this, and remember the equation is negative NFE, where F is 100, right? So then we end up with something like -2, right, times 100. So it's about -200 kilojoules, basically, right, around there. little bit higher because it's over 100% of that though, so it's gonna be like 220-ish. So, we get to the oxidation of NADH. We start with this, right? So we have the NADH goes to H plus, to this oxygen here, NAD plus and H2O. And this is about 1.14 volts. And this is enough to make 7 ATP molecules per NADH. And I say that because we know that the amount of energy required to make an ATP is around 31 kilojoules. If you just take this 21 and divide it by 30, it's around 7, right? But the thing is that one NADH only makes 2.5 moles of ATP. So what gives there? Why is it not making 7? And here is the... So from pyruvate to acetyl-CoA, right? We're going from PDH. That makes it through the last step, right? Which is E3. Then we add citric acid cycle. It adds another 8. It adds 1 ATP, adds 3 NADH and 1 FADH2. And then if you combine them all, it's going to be 1 ATP, 4 NADH, and 1 FADH2. Then we add glucose, right? So if it was total per mole of glucose, right, there's going to be 2 put in, 4 come out, so 2 net. The NADH is going to be double, right, because we're going to get some from the 2 things of pyruvate, and then we're also going to get citric acid cycle twice. So it's going to be 8 total. And then FADH2 is going to have 2 molecules of that because 1 pyruvate generates 1 FADH2 each. I wouldn't really worry too much about this. Well, if it's measuring cytoplasm, right, that NADH is going to be in the cytoplasm. But the NADH from citric acid cycle is in the citric acid cycle, right? So that's a bit weird. So the question is, if the NADHs are made outside, how do they get back in? And that's where the malate aspartate shuttle comes into play. Glycerol phosphate shuttle is true as well, but we mainly use the malate aspartate shuttle. So, for the glycerol 3-phosphate shuttle, what we start with, right, is we start with this NADH that comes from glycolysis. This then acts with glycerol 3-phosphate dehydrogenase, and what that does is it takes any dihydroxyacetone phosphate that was made in the cell, right, and turns it into glycerol 3-phosphate. This is different from glyceraldehyde 3-phosphate, by the way. Very different, right? Glycerol 3-phosphate. And so after the glycerol 3-phosphate has been produced, then we use succinate dehydrogenase complex 2 to oxidize glycerol 3-phosphate to dihydroxyacetone phosphate, and the FADH2 uses those electrons to pass them on to Q2 and do the electron transport chain cycle. Here is a massive image of the malate aspartate shuttle. Do you have to know the exact things, what's going on here? I would say no, but what you should know is that malate, right, is being exchanged with alpha-ketoglutarate. So we see here, so alpha-ketoglutarate and malate. See how they're going in opposite directions, right? So as alpha-ketoglutarate leaves, malate goes in. So essentially what's happening, right, is with this so this mechanism over here uses NADH to generate FADH2, but this method uses NADH to generate more NADH. So all we're doing is starting with malate, right? The malate goes into the cell through the alpha-ketoglutarate transporter, and as malate goes in, alpha-ketoglutarate goes out. Okay? From there, the malate is then oxidized using NAD+ into oxaloacetate, and then an aspartate aminotransferase comes along which what it does is it takes the N off of this and replaces it with the O here. And it's called aspartate aminotransferase because now the product of that reaction is an aspartate, which is able to go through this transporter as an aspartate, through the glutamate aspartate transporter. From here, we have another aminotransferase reaction where the alpha-ketoglutarate now swaps its O group with the NH2, or sorry, I mean the glutamate flops its NH2 group or NH3 plus group for the O. And what you end up with if you do that is oxaloacetate. And then from here to get back to malate, you just got to dehydrogenate it or oxidize it, which is what we're looking for in this question. Or this scenario, I guess. Here is more of the numbers, right? So we've got just a little bit more to add here. But essentially what's happening is, you know, with all of that, right? So, yeah. I wouldn't say this slide is incredibly important either, but you can probably keep it in mind just for safekeeping. And like I said before, you know, I talked about how NADH, you know, pumps from complex 1 and FADH2 pumps from complex 2. You can see here, right, that you know, NADH causes 4 H pluses to go from complex 1, 4 from complex 3, and 2 from complex 4. FADH2, though, only gets 6 H's total. So 4 from complex 3 and a couple from complex 4, right? And so if we do that, that's going to be the total amount there. So we're going to get 10 if we do NADH, and then 6 if we do complex, 6 if we do FADH2. So draw that out as 10 H pluses from NADH and 6 from FADH2. And if you do the math for this, it takes around 4 H pluses. I think I actually made a mistake in a previous lecture, but it takes 4 H pluses to produce 1 ATP. So if you just take the NADH, right, that causes 10 protons to go across, this leads to 2.5 ATP. And then if you do the same thing for FADH2, you actually get 1.5 ATP. That's where the energy loss is. And I think next time we'll talk about that a little bit more in depth. But yeah, hopefully that makes sense. A lot of moving parts in here. There's really not much I can tell you guys other than you just kind of have to memorize these. This is the part of the course where it's not the easiest to explain because there's only so much I can say other than just memorize. But what you should try and do is understand why things are the way they are. And, you know, the reason that we're able to do this whole reduction potential thing anyways, where we're dropping off electrons, is because as we go across the chain, the carriers get more and more positive, and so therefore they are more willing to be reduced. But, yeah. Hope this video was helpful. If you have any questions, feel free to stop by the IC, or leave a comment, or whatever. But other than that, thanks for watching, and happy studying.